Hey guys, welcome to the video on chapter 12, lesson number 1. Now guys, I have 3 essential questions that are going to be going into this chapter. The first one is, which experiments led to the discovery of DNA as the genetic material? What is the basic structure of DNA? What does it look like? And what is the basic structure of the eukaryotic chromosome? So this whole chapter, guys, as you can tell here, is going to focus on these 3 basic ideas. And it's all about DNA and how DNA works. Now, as a quick little side note, guys, you might notice the PowerPoints look a little bit different. That's because this time around, you can see here at the very bottom, if you look at the little pen part right here, I am using the textbooks. PowerPoint slides this time around, mostly to save myself some time because making my own PowerPoint slides takes a lot of work and effort, and I haven't had a lot of work time the last couple of weeks. So we'll see how this goes, and we'll go on from there. Okay, so first off, guys, a couple of vocabulary. The one review vocabulary you guys should hopefully know about is nucleic acid. Remember, guys, this is one of those macromolecules we talked about way back in chapter 6 last year. So it's one of the macromolecules, and its job is information storage and transfer. And you're going to find out today what that actually means. There are 2 new key terms you're going to have here pretty soon, and it's these 2 key terms right here. There's a lot more key ideas than just these ones right here, but these are the ones the book put out, and they're the ones you should probably focus on for this lesson. Okay, so first off, guys, discovery of genetic material. So, you saw that work on Mendel. You saw how Mendel learned how traits were passed on. He learned about alleles and genes and all these things, but that's just the how. That doesn't actually explain what's actually going on. So when I'm saying that an allele is being transferred from mom to dad, so if I have, say, for example, a big T and a big T, what are these big T's? I mean, what are they exactly? And we actually, when Mendel did this, we actually had no idea. We knew something was being transferred. We knew it was on the. We kind of thought that it had something to do with DNA or proteins, but we didn't actually know what was going on. We just knew that something was being transferred from parent to child that was involved in genetic information. And we weren't sure if it was DNA or proteins. Well, a couple of experiments taught us about how this worked, and you guys are going to watch these experiments because I'm going to have you guys do some research on these experiments and find out why they worked, like what actually happened, and you're going to have to explain it. So the first one was done by a guy named Griffith, and what he did is that he basically had 4 different groups. He had a bacteria. This one here is a disease-causing bacteria. If you put that disease-causing bacteria into a mouse, it kills the mouse. Okay, bummer. This bacteria kills that mouse. Well, this bacteria right here, which is very similar to this one here, but is a smooth strain, if you put it into a mouse, the mouse lives, no problem. Now, if you take the exact same evil bacteria and you put it with heat first, because if you remember, heat kills bacteria. It's why they always tell you to boil water and to do stuff like that, because boiling water kills things. And then you put that into a mouse, the mouse lives. But here's the weird thing came out. Here's what he tried out. He then took the S strain, the one that would kill you. He then injected it. He then put under heat treatment, so that would have killed the bacteria. He then injected basically the dead bacteria with the non-lethal strain that you saw right here into the mouse, and the mouse died. So somehow the living bacteria strain that was at first totally harmless to the mouse became deadly when you gave it to when you put it in combination with the deadly strain. And that's really weird because it showed that whatever's going on, somehow this bacteria right here was getting something from this bacteria right here that was causing, it caused it to become deadly. So what came out is that obviously something was going on that was causing the deadly bacteria to give something to the non-deadly bacteria. And what he concluded is that when you kill the S cells, the ones that were deadly, DNA was released when you killed that. So that made him think, maybe that's causing the DNA. Maybe the DNA is a thing that the non-deadly version is taking up, and it's going to change it into a deadly version. It's a theory, but it wasn't enough to make it conclusive, because it just shows that, well, we know DNA is released when you kill the S cells, but that doesn't mean 100% certainty that that DNA is being incorporated into the S cells. And that's where Hershey and Chase came about. Hershey and Chase, what they did was something a little more clever than this. They used what's called radioactive labeling. What this is, when you take molecules, and you might learn this in chemistry, but certain molecules like 32P, so it's a terrible 3, sorry about that, guys, and 32P, you can label that, and it's radioactive. And what that means is that you can then take like a Geiger counter and actually see if it is emitting particles. So you can actually test for radiation. So what he did was, he took one group of DNA, he labeled the DNA with phosphate through 2, and one with the proteins labeled. So here he had the DNA labeled with radioactivity, here he had the proteins labeled with radioactivity. What he then did is that we've known for a long time, these little viruses right here, we call them bacteriophages. They actually inject something into bacterial cells that causes them to reprogram. So whatever these guys are injecting into these things right here, it basically acts a lot like hereditary. It brings that hereditary material in there. We didn't know what it was. So what happened was that his control variable here was his independent variable was DNA versus protein labeled radioactively. What he found out is that when everything was said and done, and he went through the process, the radioactive part of the bacteria ended up being the DNA. Now, if you look right here, the bacterial cells, when they did their rolling after, they didn't have any of the radioactive material. So the bacteria were not radioactive when you used the protein labeling. However, they were radioactive when you used the DNA labeling. What that showed is that the virus was injecting DNA into the bacteria, not proteins. And that pretty much definitively proved that DNA was the material of heredity. Now that told us what DNA was, that it was the one doing that, but it didn't tell us anything about how it worked. And that became into the next few experiments. So what we kind of found out, guys, is that DNA is composed of monomers. If you remember the word monomer is a smaller part of a polymer, is constructed of what we call nucleotides. And nucleotides are just small subunits of nucleic acids. And they basically have a 5-carbon sugar, that's this thing right here. They have a phosphate group, well, that's this thing right here. And they have a nitrogenous base, and that's this thing right here. And every single DNA molecule has each of these 3 parts. Now, the only thing that changes from DNA molecule to DNA molecule are these things right here, what we call the bases. And there are a total of 5 bases in each for DNA. There's adenine, guanine, cytosine, and thymine. Now, uracil is a 5th one, but uracil is only found in something called RNA, and we're going to talk about that in lesson 2. So for now, just know there are 5 bases. 4 of them right here are in DNA, and 1 of them is only in RNA. So a guy named Chargaff, what he then did okay, so we now know that DNA is full of adenine, guanine, thymine, and cytosines, but what's going on? Where are these molecules there? What are they doing? So what he did is that he actually began to calculate, well, what is the percentage of each one of these compounds? And what he found out is that the A's and the T's were almost identical, and the G's and the C's were almost identical. So if you had 31.3% adenines, that means you were going to have basically almost the exact same number of thymines. What he concluded then okay, that means that the adenine and the thymines are connecting to each other. That means that these two are actually grabbing onto another. And that came out with what was called Chargaff's rule, is that the cytosine, the C's, always bond to the G's, and the thymines, they always bond to the adenines. And that gives you that showed us kind of like why these nucleotides were there and what they were doing. So the nucleotides, basically what that meant, if you look back here, is that the adenines, oh, come on, here we go. The adenines right here were always binding to the thymines, and the guanines were always bonding to the cytosines. And there was no, and it never went any other way other than that. So we learned that information. Now, the last thing, guys, came about, like, okay, so now we know that DNA is heredity. Now we know that the adenines and the thymines bind, and the guanines and the cytosines bind. The next real question was, okay, well, what does DNA look like? And that mystery was solved by these four scientists right here: Rosalind Franklin, Maurice Wilkins, Francis Crick, and James Watson. And these guys are extremely famous in biology. And I think Watson is still alive, if I'm not mistaken. I think Crick is no longer with us anymore. Neither is Franklin. But these guys right here were essential to learning this. And what they discovered, guys, is that DNA is in what's called a double helix. A double helix is that classic picture you've seen of a DNA molecule where it looks kind of like a winding strand of nucleotides that move around each other. And what ends up happening is it ends up looking, like I said, like a thing is. And using the data they got, sorry, I'll go back a second. So how they figured this out? They figured this out using something called X-ray diffraction. As you might notice, there was these are all physicists, because X-ray diffraction is a technique in physics to actually look to see what molecules look like. And what they discovered is that DNA ends up having that double helix model. And the model was essentially, it just looks like a twisted ladder. So you've probably seen these pictures before, and they're in your book. I'd highly recommend looking at them. And really, the DNA just looks like the rails of a ladder that are represented by alternating deoxyribose and phosphate. That's a fancy way of saying that essentially it's just a ladder built out of these blocks right here, where each of these blocks is going to be doing something. And if you look at this structure right here, this almost gives you an idea of kind of how it looks like. That each one of these steps right here is a rung on the ladder as you work up it. And as you would see, is that you see how here the G's and the C's, they're combined together, the A's, the T's, G's, C's, A, T. And these bind themselves into what almost looks to be this spiraling looking shape like this. And it looks almost like a giant spiral staircase. It's actually pretty cool. Now, there are two strands on a DNA molecule. There's what's called there's the top strand right here, which runs from the what's called 5' to 3', and the 5' just refers to the carbon right here, and the 3' just refers to the carbon right here. And it always runs this direction. And the other strand runs in the opposite direction. So one runs this way and one runs this way. And the two strands essentially run in opposite directions. And you might be wondering, okay, why does that matter? Who cares why these DNA strands run in opposite directions? Well, it ends up mattering quite a bit later on when we begin to use DNA replication and we begin to actually turn DNA into proteins. So for right now, guys, just know that DNA runs in the 5' to 3' direction, and the opposite strand always runs in the opposite direction. The last part, guys. Now, in prokaryotes, the DNA molecules are contained in cytoplasm, and in eukaryotes, it's in the individual chromosomes. So if you are a prokaryote, basically a bacteria, your DNA just floats around inside of the cytoplasm. If you're a eukaryote, the DNA is actually inside the chromosome. So all that stuff you learn about in mental genetics, the chromosomes, all those chromosomes, they're just DNA. So, last part, guys, before I'm done, is going to be the chromosome structure. So, the structure of chromosomes, guys, basically what it is that there are these things we call nucleosomes. And if you look right here, they're basically these little boxes. The chromosomes, if you can see right here, is basically one giant winding circle of DNA. And these winding circles of DNA are built out of these little building blocks that are called nucleosomes. And nucleosomes are basically just the building blocks of DNA. And really all they are is they're just DNA plus histones gives you a nucleosome. And it's just a way to give it a building block. So each nucleosome basically looks like a block like this, and you can have block upon block upon block, just like this, and it lets you build that spiral structure that you see right here. And that's it, guys. So what I want you guys to do is I want you guys to attempt to answer these 3 questions in your journal. There's no, because remember, guys, because with these questions here, I don't really care about if you're right or wrong. I just want you to give it a try, see if you can't pull it off. And these are the 2 key terms for this particular chapter. Thank you for watching, guys, and I hope you guys have an awesome day.